Hello again, everyone. Thank you for joining us today. So we'll start our webinar. So I'll share the presentation. For today's webinar around profiling pharmacokinetics in a microfluidic platform. So we have today Benoît Maisonneuve, our biopharma and clinical translation project owner, and Tudor Petris, our biopharma senior scientist, who is leading our R&D development of PNS and pain platforms. To give you a quick overview of today's program, so we'll first present to you who we are as Netri and the solutions that we have today. Then we'll get into how to mimic PK profiles and therapeutical schedules for anti-cancer drugs in Netri devices. And we'll end with a Q&A session. So if you have any questions during the webinar, do not hesitate to write them down in the chat section, and we will get to them at the end of the webinar. So who we are today as Netri. So we were founded back in 2018 by Thibaut Neger and Florian Laramendi. Today we are 35 collaborators from multidisciplinary backgrounds. We have more than 100 clients all over the world, both from pharmaceutical companies, but also academics. And we have 16 patents on our technology. And so the focus, the base of our what we do at Netri is neurons. And we use these neurons to actually record biological neural connections to digitally assess drugs, compounds, by using neuroprofiling. So we know that neurons, they convey electrical signals. And so in that way, we can, depending on the stimuli and the state of the neuron, the message can be seen as a code or combination of zeros and ones. And in that way, the neuron is not just a biological entity, but also a sensor that is capable to digitalize human biology. Not only that, neurons are sophisticated sensors. They host a variety of dedicated receptors that, depending on the compound, whether the receptors are activated or inhibited, they can encode a complex response to the stimuli. And this response can be deciphered when we look at different parameters of the electrophysiological response. Then this response to a specific compound can be specific. Or just to simplify, depending on the compound or condition applied to the neuron, we can generate a digital signature. This digital signature can be compared to a library of compounds and known conditions that have a specific efficacy or safety. And then we can predict that compound's safety, efficacy, but also a diagnosis. So to do that, we have developed a neurofluidics NEA hardware, which is the world's first high-throughput compartmentalized electrophysiology platform that combines our microfluidic chips with NEA technology. And so, as you can see on the right, so you have an example of a device where you have 3 channels, with the middle channel here being having the role of a microfluidic barrier. So when we culture neurons, when we put neurons in, for example, the left channel, during the culture, the cell body or the somas will stay in the channel where they were seated. So that's the spine compartment. And then the axons will grow and innervate a particular organ or target organ in the channel 3. And in that channel, we can actually apply different compounds or different conditions, and then record how the neurons respond or the message that is vehiculated by the neurons via microelectrodes. So we use this platform in several programs, so mainly in pain management, quality of life, food environment safety for the neurotoxicology axis, and dermatocosmetics. And in parallel, we have we're doubling in vitro diagnosis with a focus on neurological troubles. And so now I will let Tridor take the lead and present the PK profiling.
Hi everyone, thanks for attending our webinar. So we are just starting with a brief introduction about the main anti-cancer therapies, which are currently rely on biochemical methods like chemotherapy, hormone therapy, immunotherapy, stem cell transplant, or targeted therapy, which is using nanotechnologies. Also, we can use physical methods as surgery, radiation therapy, hyperthermia, or photodynamic therapy. But also new therapies are at all time being introduced using antibody-drug conjugates, tumor-infiltrating activated lymphocytes, and also inhibitors of DNA damage response pathways as PARP, ATM, and ATR inhibitors. The administered anti-cancer therapies are being evaluated according to efficacy and adverse effects, and all these are depending on administration, distribution, metabolism, and excretion of these drugs. And thus, in the whole process of drug discovery, from basic research to clinical trial, passing through in vivo assessment with using animal models, an important stage is to define a drug pharmacokinetic profile. So understanding and being able to mimic the PK profile for anti-cancer drugs is a critical step in order to approve and a drug and for the patient output as well. So there is a constant urge to define and understand the PK profiles. These PK profiles can be simulated in silico using in vitro, in vivo extrapolation methods, or it can be explored in animal models, but here we might retrieve interspecies PK variability, and also we can have ethical considerations to take care of. So thus the organ-on-chip developers started to explore this ADME in vitro platforms. Currently, an anti-cancer drug PK profile can be simulated in silico, then can be assessed in vitro, but only sequentially and using multiple bolus concentrations corresponding to different time points, and this is not exactly a necessary physiological way to do it. Then the best results are being selected and passed for in vivo model, and then they are entering the early clinical phase trials where the most failures are being observed. And thus, we are introducing an in vitro approach to mimic any kind of PK profile and any kind of therapeutic schedule for anti-cancer therapies. Our method is correlating data from in silico, in vitro, or in vivo, and clinical trials data. Our devices can be supported by a Beckman Biomac i7 automated system, which can mimic any type of PK profiles for one or multiple drugs, and followed by a customized therapeutic schedule. For this purpose, we can use our pumpless and easy-to-use platforms, either neurofluidics MEA platforms or multifluidic platforms. Thus, instead of using a classical bolus dose, we are mimicking PK profiles using a sequence of addition and removal of specific drug concentration, each corresponding to a point on the PK profile curve. And then these PK profiles can be repeated many times in a therapeutical schedule using programmable time intervals from 1 to 12 hours, but trying obviously to avoid excessive mechanical stress. Also, we can set customizable treatment schedules that might last for various time intervals from 7 to 21 days or more, but this depends definitely on the cellular model, if it is 2D or 3D, for example. We are able to set monotherapies or combination therapies, as you can see in this diagram, spaced by washout intervals. We can also use our open or semi-open architectures to perform time point sample collection or dynamic or endpoint assays. As an example from a previous life, which you can find in this publication linked here, you can observe an in vitro colorectal cancer 3D model treated by this irinotecan metabolite, SN38, either in a monotherapy or combination therapy with an ATM inhibitor. The treatment efficacy was compared afterwards according to the treatment schedule and in vitro treatment setup, a static bolus versus microfluidic pharmacokinetic profile. And the results, as you can observe in the paper, show the advantages of using microfluidic PK setup and its ability to mimic and to superpose results with in vivo animal models, and thus avoiding, obviously, the static setup over prediction. Also, the proposed workflow is pretty straightforward. The pharmacokinetic profile is automatically generated with high accuracy, and the data are being confirmed by mass spectrometry. Pharmacokinetic profile can be automatically customized based on in silico, in vivo, or clinical data using our robot. The same platform performs automatic mixture for compounds to be delivered and the customized therapeutic schedule and duration. And also, this platform is automatically distributing the therapies to the tumor-on-chip model. And as a conclusion, our platform and associated protocols offer technical advantages as it is pumpless, it is mixerless, is autonomous and modular. It is able to deliver therapies according to the customized PK profile and therapeutic schedules, including washouts. And also, it is working for monotherapies and combination of drugs and methods. The advantages over the therapeutical model includes the capability to explore single drug therapies, but also combination therapies that will become extremely costly on animal models. For example, a therapeutic schedule to include 3 drugs will require monotherapy, then combination of paired drugs, and finally, triple combination of drugs. This is becoming extremely costly and using massive amount of cells to develop tumor in animal models. And also, there is the problem that animal control groups do not always survive long-term experiments, contrary to 3D patient-derived organoids. So regarding these advantages and the data we have introduced to you, I would like to thank you for your attention and for participating to our interesting webinar. And also, we are definitely looking forward for the questions.
Yeah. So thank you, Tudor. So if you have any questions, it's the time for us to answer those. So you can write them down in the chat section. Okay, so we have a first question. What would be the throughput number of conditions for a 4-week experiment using PK-related anti-cancer therapies?
Oh, yeah, well, that's a good and interesting question. It's a challenging question, actually, to calculate, like, on top of my head. I can tell you that we have performed already a 4-week experiment using, starting with 4 conditions, each therapeutic condition using its own PK profile, and but it can be scaled up to 10 conditions at a time. And running for 4 weeks, this might be a really great opportunity for scalability regarding this kind of experiments.
Okay, thank you, Tudor. Let me see, do you have any questions? Okay, we have another one for Tudor. Is the number of points to be reproduced on the PK profile, is variable reduced to 8 points or flexible?
It is flexible, definitely. Regarding the PK profile you want to define, because as you have seen in the animation I have displayed, 8 points seem to be like a reasonable amount of points in order to reproduce correctly the smoothness of the PK curve. But also, we don't want to perform excessive stress to the biological model. So less than 8 points will mean that we are changing media every less than 3 hours at a time, and this might induce an excessive mechanical stress on the tumor model. We very well know that tumor models and extracellular metrics, if used, is being very sensitive to mechanical stress, and that might definitely influence the output of the treatment. And to move away from the physiological reasoning for the treatment. So I suggest a maximum of 8 points, and minimum literally depends on the PK profile you want to simulate.
Okay. Thank you. Hopefully we answered your question. Okay, we have one maybe for Benoit. Is Network Platform capable of accommodating metastatic models to assess similar PK-dependent therapeutic schedules?
Yes, absolutely. We can do that, not by using, I guess, the device that we showed here, that is more adapted for cells population when they are going in 2D, but you can actually use our duplex multifluidics, the well one, that can accommodate three-dimensional structures of any kinds. You could definitely start to use these kind of models. The interesting thing with the method that Tudor presented is that it's not necessarily linked to a specific architecture. Mm-hmm. It really is a way to approach these kind of questions, and that can be adapted to pretty much any kind of architecture we have installed, basically.
Okay, thank you. Okay, let's check for some questions. Do we have more questions? Okay, we have one. Is the lack of continuous flow influencing the biological model response to treatment?
That's a tricky question. Thank you. Everyone is asking if this kind of therapy, this kind of approach can be influenced by the lack of flow. And I will say not really, because the tumors are they have a very particular blood flow, literally. And the blood flow in tumors can vary from 0 to 100 microliters per minute. So we can have very intense blood flow or literally blood stalling in the tumor. So as like a decent physiological compromise in simulating this model, changing media every 3 hours ensures enough media change and definitely mechanical stress to mimic properly the treatment and the output. And the results we have had in experiments compared to in vivo data showed us that the model is really reliable, and the way to treat it works pretty well.
Okay, thanks. Okay. Okay, another one, probably for Tudor. Yes, again. Is it possible to assess complex 3D gel, hydrogel encapsulated tumoroids or PDOS?
As a working principle, yes. It really depends on which kind of platform from the laboratory devices you want to use. Encapsulating cells in hydrogels in order to perform organoids can also work as well, but also it works for PDOs and for spheroids, tumoroids actually. And they can be encapsulated in gels exposed to the treatment in our open architecture, like the duplex multifluidics, in which we have two compartments, an upper compartment in which you can place the single or multiple tumoroids, or actually you can set the number of tumoroids you want to treat or to observe. And you can dispose them in a hydrogel, you can suspend them in a hydrogel, and definitely you have an underneath channel, which can literally perfuse and feed or treat the tumor by diffusion, and you can literally add a vascular layer, an endothelial layer, to simulate the vascular compound. So it literally, we have some very flexible configurations that can be addressed, and yes, it might work with 2D and 3D models as well.
Okay, thank you. Okay, so we discussed tumoroids, but is the current platform available for non-oncologic models and therapies based on PK profiles?
Yes, absolutely. So it's actually pretty much linked to what we said before, the few last questions I get that Tudor and I answered. As we said, this is a method that we're using, and the PK profile, like PK profiling and their importance, is not something that is unique to the oncology space. It's something that is important for any kind of drug delivery or pharmaceutical treatments. So yes, it can absolutely be used for any other disease models that you may have or questions. Once again, because this is a method, it can be used with whatever devices that we have in store, and then we just need to adapt the biological source to model the pathology that you have in mind, but in no way is it specific to oncology.
Thank you, Benoit. Okay, so maybe you can answer one last question. Is it possible to perform real-time sample collection?
Oh, only difficult questions for me today.
Sorry to
but no, it's okay. It's okay. I think I'm ready to answer this question. Yeah, definitely you can collect, you can perform time point sample collection regarding the media, but also it is possible in the future to optimize the model from the 3D model from duplex in order to perform like micro biopsies using an automated system that can literally perform this kind of procedure without disturbing or without influencing the behavior of the biological setup. But definitely we can collect samples, and this is also true for non-oncological models, especially if you consider like a 3D model of liver or some 3D structure that requires literally assessing continuously the biological behavior, so the metabolites production or the literally the health status of the biological model. So yes, it is possible and optimizable.
Yeah, thank you, Tudor.
And just to add a little bit on what we actually discussed in the last few questions, so we have other actually platforms that we're using for specific applications. So using PK profile modelization, you can of course look at efficacy. What is often done actually when you look at the state of tumoroids or tumor cells in vitro. But you can also explore all the adverse events or the secondary effects that you may have as well. That can be actually linked to the treatment schedule. So you can actually look at if the treatment schedule, if you modify it, you may potentially reduce the adverse events potential effects or just happening. But as well, like Tudor mentioned during the presentation, you can also explore a combination of therapeutics. So you can look at a combination that are used to increase efficacy, of course, but you can also think about starting to stimulate the combination that are sometimes used clinically in order to try to alleviate some of the side effects that you may have clinically. And that's especially true in some oncology context, for example. So that's also something that you can use, you can address using this kind of approach. So it's why it's really important to understand that it really is just a global method on how we can expose a biological material in vitro to a certain amount of drugs that can change throughout the day, but also throughout weeks and days and weeks, basically.
Yeah.
And also, if I may add,
sure,
we can use humanized PK,
absolutely,
which is really very important because it literally avoids the errors induced by animal PKs, which are not applicable, translatable to clinical trials.
Yeah, as soon as you have PK data from patients, either from phase 1 clinical trial, phase 2, or like depending on where you get it from, we can definitely use that as a base to work on, definitely.
Okay. Okay, so so I think there is no more questions. If you have any questions regarding what Tudor presented today, what we discussed in terms of 2D or even 3D culture models, do not hesitate to contact us. So you have our email address, contact@netri.com, or via our website. So thank you again, everyone. Thank you, Tudor. Thank you, Benoit, and have a nice day. Bye-bye.
Thank you. See you.
Bye-bye.